announcement. There will be a biology colloquium today on aggression, infidelity, and anti-image in an Australian songbird. I'm going to learn about these things in songbirds. Birds obviously have color vision. They wouldn't develop such colorful plumage if they didn't have color vision. So the things that we'll be talking about today in anatomy and physiology will not be discussed in the seminar, but obviously you can apply your knowledge about color vision to be thinking about using color in sexual selection in a bird. Another announcement is a reminder that the problem set is due on Sunday. So our goals for today are to finish the discussion of the visual system. We started that with some anatomy, and today we'll be doing mostly physiology. Most important to understand how your retina converts photons of light into electrical signals that our brain can interpret as a representation of the view that is coming into our eyeballs. So we'll understand phototransduction by the rods and cones. We'll discuss the cellular organization of the retina and how lateral inhibition is important in the retina. We'll talk about a specific set of the neurons that take information from the retina to the brain, so-called on-center cells and off-center ganglion cells, and then we'll go on to some anatomy of how that information is processed in the brain. And I hope there's time where I can introduce the auditory system, which is where we're going next. So let's start. Let's wake up with a clicker. The clickers are outside the room today, and so is the handout, just in case you missed. If you didn't come in this door, you missed picking up the handout and the clicker. Let's just start with a clicker from Tuesday. You're focusing on a nearby object in bright light. Which muscles are contracting? I'm just going to give you hints because these are pretty straightforward. I'm sorry, two choices. Well, there's three choices. There's A, B, and both. Right? Three choices. Both. Bright light means that the iris sphincter is contracting in order to reduce the amount of light coming into the retina. And nearby focus requires constriction of the ciliary muscle so that the lens is not being stretched by the zonular fibers so that it can round up and have a greater curvature to allow it to focus the near image, the image from the near object, on the retina. Okay, what nerves are firing? Action potentials to create this condition in the eye. This condition being constriction of the iris sphincter and constriction of the ciliary muscle. In other words, which nerves innervate these muscles? Motor, because all of it. Okay? You're focusing on a nearby object in dim light. You can demonstrate it. Focus on a nearby object in dim light. Too dim, but your eyes are adjusting now. You're focusing on a nearby object in dim light. Which muscles are contracting? Now you're dilating the iris in order to maximize the amount of light coming in this dim light, but you still need to constrict the ciliary muscle in order to allow the lens to round up, to allow it to focus on a near object. What nerves are firing to create this condition? It's the same, the oculomotor nerve. Okay, so these are the sort of things, I think you'll be using some of this in the problem set. This is the sort of information that will be necessary for the next exam, which is not coming up till the end of March, right? Yeah. And remember, and this is my reminder that there's a bunch of clickers in the PowerPoint from Tuesday that you can use to review. So now we're going to talk about phototransduction, transducing photons into electrical signals in the retina. Let's get oriented. So here's a cartoon of the eyeball in a mid-sagittal section, vertical section, so cornea, anterior chamber, pupil, lens, vitreous humor in the posterior chamber, retina on the back of the eye, the posterior surface of the eyeball. Okay, so we're zooming in to this area right here. So here is a cartoon of the cell organization of the retina. This is the front, so this is what's facing the vitreous humor. And this is the back, or posterior. So here is the back of the retina, and then there's the pigment epithelium, and the choroid, and behind the choroid is the sclera, the white connective tissue on the outside of the eyeball. So this is the front, and this is the back. And it's confusing because phototransduction starts in the back even though light comes from the front. Okay? So light goes through layers of cells that are relatively transparent. First they go through, the light has to go through cells in the cornea that are transparent, has to go through cells in the lens, it's cellular, that are transparent, and then it has to go through layers of cells of the retina before it is actually transduced into an electrical signal by the rods and cones at the back of the retinal nerve cell layer. Rods and cones are named for their shape, rods, cones. Rods are important for transducing dim light. Cones are important for transducing bright light and giving us color vision. If we only had rods, we would see in black and white. So the rods and the cones are the phototransducing cells. They are what turn photons into electrical signals, and we'll get to that in a few minutes. Then the electrical information is processed towards the front. So these are synapses with bipolar cells, and then synapses with ganglion cells that have the axons that go out and form, these axons form the optic nerve. So the optic nerve that you'll see in lab either tonight or on Tuesday is formed by the myelinated axons of retinal ganglion cells. So there's a through pathway, and then there's lateral inhibition. The through pathway is rods and cones transduce, synaptic information is passed on to bipolar cells. Bipolar cells process that information. Synaptic transmission to ganglion cells. Ganglion cells either increase or decrease, depending on whether they're on or off center, depending on where the image is relative to that ganglion, relative to that rod and cone. These ganglion cells fire the action potentials that take the information out the optic nerve to the lateral geniculate nucleus. So the through pathway, the straight pathway is rods and cones, bipolar cell, ganglion cell. Lateral inhibition is mediated by interneurons that have names in the retina. And the names of the interneurons in the retina are horizontal cells at the first, that do lateral inhibition at the first synapse, and amacrine cells that do lateral inhibition at the second synapse. What's the benefit of lateral inhibition? Sharpening visual fields. Allows us to see with fine detail. Here is a micrograph of a human retina going through. So let's get oriented. Here is the front of the eye. Here's the vitreous humor, the anterior surface of the retina. So this is the anterior surface of the retina. Here are the ganglion cells. We're seeing their cell bodies. We're not seeing their axons. Here is the layer. It's called the plexiform layer. I'm not going to hold you to any of this nomenclature. The inner plexiform layer is the area where this set of synapses, inner plexiform layer is where this set of synapses is. So you can see it's not trivial. It's a major synaptic connection, thick with lots of synapses. Then the inner nuclear layer is where the cell bodies of the bipolar cells and let's get oriented. The inner nuclear layer is cell bodies. That's why it's purple circles. Cell bodies of the bipolar cells, the amacrine cells, and the horizontal cells in the inner nuclear layer. Then a layer of synapses. The outer nuclear layer is the cell bodies of the rods and cones. And then we have the outer segments of the rods and cones. So cell bodies, and then the outer segments of the rods and cones. It's the outer segments that do the first step of phototransduction. They are what absorb photons and turn it into something electrical. So you can see it's a well-organized, layered structure. We see this again in the brain, a lot in the brain. Cell bodies, synaptic layers, rods and outer segments. Then here's the pigment epithelium, the choroid, and the sclera, the white of the eye. Remember, this is the back, this is the front. This is where phototransduction happens. This is where we're focusing now. Just a tidbit of information. Your retina is not uniform. It's not the same. So let me just put up a cartoon. The retina is not uniform. There is a spot on it called the fovea, which is where we have our highest visual acuity and where we have our best color vision. So when we are putting our focus on a spot, we are trying to place the image of that spot on our fovea. And the rest of the image is getting put on the peripheral part of the retina. So I'm staring at RJ's face, I'm putting the image of his eye on my fovea. And everything else around here I can see, but I can't see very sharply because this is not on my fovea, whereas the image of RJ's face is on my fovea. At the fovea, let's not worry about the angle, but at the fovea, we have the highest concentration, the blue line, the highest concentration of cones, and a relatively low concentration of rods, because our goal there is acuity and color. As we go away from the fovea, the percentage, the number of cones goes down, and the number of rods goes up. So we have more rods in our peripheral vision and more cones in our foveal vision. We see better, in bright light, we see better best with our fovea. In really, really dim light, where the rods are more sensitive than the cones, we actually see better in our peripheral vision than we do on our fovea. Some of you who are stargazers might know that if you go out at night, you let your eyes adapt to darkness, and you're looking at a really, really dim star, and you stare at it, you can't see it. But if you move your eyes just a little bit to the side of that really, really dim star, you can see it in your peripheral vision, because you're putting your rods, not instead of your cones, on that very, very dim star. Anyway, rods are outside the fovea, black and white vision, high sensitivity. Cones on the fovea, color vision, low sensitivity, but high acuity. There is a blind spot. There is a blind spot, which is where all of the axons from those ganglion cells have to go out the back. So all these ganglion cells on the inner surface of the yellow of the retina are going out the optic nerve. So there's a spot right here where there are no rods and cones. There are only axons of the ganglion cells that are collected here to go out the back. And so that's the blind spot. So the blind spot is where the optic nerve is. That's not where the fovea is. The fovea is a few degrees away from the blind spot. Okay, so now we're going to focus on the rods and cones and how they convert light into vision, light into electrical signals. Outer segment, inner segment. The inner segment is where the cell body is, all the stuff you learned about in CGI, the expression, DNA, RNA, expression of proteins. But the outer segment is where phototransduction happens in both rods and cones. So outer segment, inner segment, and then the synaptic area. Synapses here with bipolar cells in the through pathway. The rods are specialized with these intracellular stacks of membrane called discs. The cones are specialized not with discs, but with this sort of cone-like structure, tooth structure. But the similarity is that there is abundant membrane for the expression of the receptor that receives the photons. In this slide, rhodopsin is named because rhodopsin is the name of the photoreceptor in rods. Rhodopsin. Rhodopsin is the name of the protein that receives photons. In cones, we're going to have the names of other opsins. This slide uses pigment molecules. Most people call them opsins. So rhodopsin is the opsin in rods, and then we're going to have other opsins in cones. Opsins are GPCRs, G-protein coupled receptors. So you know all about this. I don't have to go through it in great detail because you can apply everything you learned about GS, GI, and GQ. Well, specifically, we're going to be focusing more on, the things you learned about those G proteins, GPCRs, can be applied here. So here's the GPCR in the membrane. 7, I can't count them, but 7 transmembrane helices in this stick model of the GPCR. Here is a ribbon diagram of the alpha and the beta gamma subunits of the G protein. So here's the receptor, here's the G protein. In the visual system, the G protein has a name, and it's called transducin. Transducin is the name of the G protein. In this case, opsin is the name of the receptor. And if we're talking about a rod, we're talking about rhodopsin. Palmitate, myristalate, farnesylate. These are the lipid linkages that tie the alpha and the beta gamma subunit that embed those proteins, not embed them, but couple them to the membrane. So the GPCR is in the disc membrane, or the tooth, the comb membrane, and the alpha and beta gamma subunits of the G protein are tethered to the membrane. The ligand is not the photon. The photon is not really the ligand that activates the receptor. There is an endogenous ligand that is part of the protein. Well, it's not really part of the protein. It is bound to the opsin, and it's called retinal. Vitamin D. No, vitamin A. Retinal. Retinal is a small molecule that is the ligand that activates the GPCR when light hits it and does a photoisomerization, a cis to trans isomerization of the retinal is what converts, is the ligand that activates the GPCR. So a photon hits the retinal, conformational change, cis to trans isomerization, conformational change of the G protein, no, conformational change of the receptor, conformational change of the G protein, GDP dissociation, GTP association, alpha separating from beta gamma, alpha separating from receptor, same story that you learned about for the other G proteins. Don't forget about GTP here and conformational change. I ask you to tell me the story of phototransduction. Yes?
Which G pathway is this?
You'll see in a moment. It is none of the, it's none of the above. So now let's talk, before we answer John's question, let's talk about the differences in the different opsins. Rhodopsin, so what we're calling here is the response as a percent of maximum, the response of the opsin as a function of the wavelength of light. Purple light, ultraviolet light that we're blind to down here, purple light, blue, green, yellow, the rainbow, red light, infrared light up here that we're blind to. This is our visual spectrum. Rhodopsin has a peak in the blue-green light. So we are most sensitive in dim light to blue-green light. Because that's what our rods are seeing. The opsins in our cones, we have trichromatic color vision, except for a few mutant humans who have tetrachromatic color vision. We have three different color sensors, three different opsins. We have blue opsins, green opsins, and red opsins. Of course, the opsins are not those colors. Those are the colors that those opsins have been tuned to be activated by. So the opsins in the blue cones, or the blue sensitive opsins, have a peak in the pretty deep blue violet. The opsins that detect, we call these the green opsins because they are maximally activated by green light. We have opsins that we call the red opsins, but they are really maximally activated by yellow light. But we call them red because they're the only ones that actually give us any information about red. We call them red opsins or red cones, but they're really most tuned to seeing yellow. The different opsins, the thing that makes them different are single amino acid changes, mutations, in the GPCR, in the opsin. Retinal is the same for all of them. So the thing that actually gets converted by the light is the same. What has changed is how that retinal is in, what that retinal is embedded in. And there's just a dozen or so single amino acid changes between rhodopsin and the specific color opsins that give them these differences in their peak visual detection. So you can see rhodopsin and blue opsin, there are wavelengths that activate both. Green opsin and red opsin, there are wavelengths that activate both. But red opsin, if you go all the way out to very red light, only the red opsin detects it, because only the red opsin undergoes the conformational changes when that red light hits that retina. Main point, small differences in the opsins. Small, just a few amino acid differences in the opsins give us our range of color vision. Insects, some insects can see in the infrared, some insects can see in the ultraviolet because of different mutations in their opsins. What we call infrared is just because of the way our vision has evolved. If we were insects, we would call this red or something. We wouldn't call it infrared. It's hard language. How do you describe red to something that doesn't see red? I mean, how would an insect describe what they see when they're seeing a wavelength out here to us? We have no basis for knowing what that is. So now, let's get to John's question. What pathway is this? And it's a little bit weird. So here we are. Is this a rod or a cone? It's a cone. Yeah, it's got this serrated edge structure. This is where the opsin and the G protein are highly expressed. So here, this is just a zoom-in cartoon of that membrane. Here is the opsin. Here's the retinal. Here's transducin. Okay, so this is the G protein story. But in order to tell the G protein story, I have to back up. And so this is what happens when light hits that opsin. It's that part of that cone. But I have to back up because we have to tell what happened in the dark before the light hit that retinal. In the dark, in these cells, there is an enzyme called guanylyl cyclase that is working all the time. It's just cranking out cyclic GMP. So just like adenylyl cyclase makes cyclic AMP, guanylyl cyclase makes cyclic GMP from GTP. It's working all the time. And in the dark, cyclic GMP accumulates. Cyclic GMP is a ligand for a ligand-gated cation channel. So in the dark, with a lot of cyclic GMP, there is a lot of opening of this cation channel. What does that do? What does darkness do to the resting membrane potential of this cell relative to what you think of as normal? Is it the same as normal? Is it hyperpolarized relative to normal? Or is it depolarized relative to normal? Depolarized relative to normal because we have this large cyclic GMP-gated influx of sodium and calcium. This influx of cations depolarizes this cell in the dark. There is a large, the biophysicists call this the dark current. the current through these channels in the dark. Cyclic GMP dependent. Now, let there be light. Light hits the retinal, conformational change of the opsin, conformational change of the G protein called transducin. What's missing here? GTP or GDP dissociation, GTP binding. So the same GTP that guanylyl cyclase uses to make cyclic is there to activate, not shown. So annotate your image or make note. GTP is necessary for the activation of transducin. The activated alpha subunit of transducin, GTP liganded transducin, activates an enzyme called phosphodiesterase. So it's not GS, and it's not GQ, and it's not GI, it's transducin. It activates phosphodiesterase. Phosphodiesterase, as its name suggests, breaks cyclic G down into just ordinary GMP, phosphodiesterase. It cuts cyclic G. So in the light, breakdown of cyclic GMP, decrease in this concentration of cyclic G, closure of the cyclic G-dependent cation channel, what's that going to do to this membrane potential? Hyperpolarize it relative to its dark level. So light, this is the weird one, won't be the last time I say weird, because this system, light causes a hyperpolarization of the photoreceptor cell. But that's an electrical signal. Right? An electrical signal could either be a depolarization or a hyperpolarization. We have just converted light, a photon of light, into a hyperpolarization of the membrane potential. In the dark, it was relatively depolarized, and then light causes this cell to hyperpolarize. There is the conversion of a photon of light into an electrical signal. We haven't finished yet, but we have gotten started. Any questions? Yes?
So there isn't necessarily like a resting, like neutral, it's either pretty much like in the dark it's depolarized relative to the light?
In the dark, so if you consider darkness resting, then you would say that the cells have a resting membrane potential that is relatively depolarized compared to other normal cell resting membrane potential. Let me just show you. So in the darkness, these cells have a resting membrane potential of around minus 35 millivolts, instead of what you're normally thinking of as for the typical neuron. This isn't a neuron, this is a photoreceptor cell, but this cell's resting membrane potential is minus 35 millivolts. It's depolarized compared to what you consider normal in the dark. So in the darkness, the resting membrane potential of these cells is negative 35. When a photon of light hits it and activates that pathway, closing the cyclic GMP gated ion channel, it causes a hyperpolarization. There it is. It goes from minus 35 to minus 55. This is actual data from a photoreceptor cell. Weak light causes a weak hyperpolarization. Brighter light, or a light that is more aligned with the spectrum of maximal activation of that opsin, causes a greater hyperpolarization. This is a graded response. This is not an action potential. This is a graded receptor potential. Small, medium, large, everything in between. Receptor potential in response to light. So light hyperpolarizes the receptor cell. Questions? Now we have to go on to the next step, which is neurotransmitter release. And as you would expect, now things get a little normal. A hyperpolarization of the cell causes a decrease in neurotransmitter release from the receptor cell. So in the dark, relatively depolarized, there is a lot of dark neurotransmitter release. I mean, the neurotransmitter is not dark, but in the darkness, this cell is releasing neurotransmitter to trigger changes in the downstream cells. In the dark, this cell is releasing neurotransmitter. In the light, the hyperpolarization closes voltage-gated calcium channels, causes a decrease in neurotransmitter release. So in the dark, there is a lot of neurotransmitter release. These neurons are, these photoreceptor cells are specialized to be able to release a lot of neurotransmitter all the time. Well, all the time in the dark. They actually, the name of the synapse, there's I think a slide that shows it here. The synapse has a special name. It's called the ribbon synapse because these structures here help it release a lot of neurotransmitter in the dark. I won't hold you to that name. In the dark, there's a lot of neurotransmitter release from the photoreceptor cells. In the light, the hyperpolarization decreases neurotransmitter release. That's a change, right? So you're used to thinking that activation of a signal should be an increase in neurotransmitter release. But decrease in neurotransmitter release is a change. So we have seen an electrochemical change in the photoreceptor cell in response to light. It's a little counterintuitive. It's not the way if you were to have told me how you wanted the system to work, it's not what you would have come up with, but this is the way evolution has come up with photoreception. John?
How do rods work better in the dark when they're relatively depolarized?
So all the cells are relatively depolarized.
Yeah.
The rods work better in the dark because they're rhodopsin. Because their rhodopsin is more sensitive to light than are the color opsids. So this slide suggests that they all have the same maximum, but this is relative to their own maximum. Rhodopsin would actually be through the ceiling if we put them all on the same scale. We're putting them on a scale here where every opsin is getting to its own maximum. If we were to put this on a regular scale, the reds would be low and the rhodopsins would be high because they are more sensitive to light. Okay, so now we have converted a photon, small number of photons actually, into an electrochemical change in the receptor cell. So now I want to put this together into understanding how the ganglion cells, so I've skipped a couple of cells, I've skipped the bipolar cell, just for a moment. But the information that comes from the retina and goes to the brain are action potentials. And on this cartoon, these spikes are supposed to illustrate the frequency of action potentials from a retinal ganglion cell. Okay, so I'm trying to explain an interesting phenomenon. Some ganglion cells increase their action potential frequency when the photoreceptor that is connected to them is illuminated by light. In some retinal ganglion cells are, let me just get oriented here. Some retinal ganglion cells are actually inhibited by a spot of light. So if a spot of light is hitting an on-center ganglion cell, it is activated. Its action potential frequency is activated by that spot of light. And if a spot of light is hitting an off-center ganglion cell, its action potential frequency is decreased. But remember, electrical information can be carried both by an increase in action potential frequency and by a decrease in action potential frequency. Both of these ganglion cells are carrying information from the retina to the brain. So now let's try to figure out, how does a spot of light cause these different forms of electrical signaling in the retinal ganglion cell? And the strict name for these cells is on-center, off-surround, or off-center, on-surround. But we're going to just focus on the center. On-center and off-center. Let's not worry about the surround. Here's an off-center. Here's an off-center cell. Cells turned off. Less action potential by the spot of light on their center. Off-center. Okay, so let me go back. An on-center cell, an off-center cell. Let's not worry about the surround. Okay, so here's how it works. So your handout has this slide, but it's all labeled at one time. So this slide is going to walk us through this. So my recommendation, my strong recommendation is, when you're trying to learn this mechanism, use the PowerPoint on Moodle to walk through it. Don't use the static printout, because that doesn't illuminate, that doesn't shed light, whatever. It doesn't tell us the story as much as walking through it. Okay, so here's a photoreceptor cell. It's a cone. It's connected to two different bipolar cells, which are connected to two different ganglion cells. And this is simplified, but it's not oversimplified. Well, it's a little oversimplified. So let's walk through it. Light hits the photoreceptor cell. It causes, well, okay, we put an electrode in this photoreceptor cell so that we can measure its response. Light, that's the yellow bar, light causes a hyperpolarization. Right? Just by the mechanisms that we had up on the slide a few minutes ago. Light causes a hyperpolarization. Now it gets complicated. So now you have to wake up. The two different types of bipolar cell have two different types of neurotransmitter receptors. The neurotransmitter that is released by the photoreceptor cell is glutamate. There are excitatory glutamate receptors on some of the bipolar cells and inhibitory glutamate receptors on other bipolar cells. So now, in the darkness, there was a lot of neurotransmitter release that caused a lot of depolarization. Let me just walk through this. So this is now in the dark. We've now hit this photoreceptor cell with light. The amount of neurotransmitter released from this cell goes down. There was a lot of glutamate being released in the dark, and now there is less glutamate released in the light. Let's record from this one. This is an inhibitory, this is a photoreceptor, no, this is a bipolar cell that expresses an inhibitory glutamate receptor. Let's see if you can predict this. Decreased release of glutamate is going to cause what electrical change in this cell? Decreased glutamate that activates, the glutamate activates an inhibitory receptor. Decreased inhibition is going to cause a depolarization of this bipolar cell. Decreased amount of neurotransmitter, so decreased inhibition allows it to depolarize. Now you can predict already what's going to happen to this one. Decreased glutamate, decreased activation is going to cause a hyperpolarization of this cell. So some bipolar cells are depolarized by the light that hits their photoreceptor cell, the photoreceptor cell to which they are coupled. And some bipolar cells are hyperpolarized by the light that hits their coupled photoreceptor cell. Now it gets simple. What happens to the amount of neurotransmitter that is released from a cell that is depolarized by light? Increases. Depolarization, increased voltage-gated calcium channels, calcium influx, synaptotagmins, snares, increased neurotransmitter release in the cell that is depolarized by light. So now, what is going to happen to the action potential frequency in this ganglion cell that is depolarized? Oh, I forgot to tell you. This is glutamate again, an excitatory neurotransmitter. So what's going to happen to this cell? Is it going to depolarize or hyperpolarize? Depolarize, and what's that going to do to the action potential frequency in this ganglion cell? Increases. Light caused an increase in action potential frequency in this population of ganglion cells because of its coupling to this type of bipolar cell. And this was the bipolar cell that was expressing the inhibitory glutamate receptor. So light inhibition of inhibition leads to an on-center response. The other type of ganglion cell is inhibited by light. Light, inhibition of depolarization, less neurotransmitter, less excitatory neurotransmitter release, less glutamate release, less activation of this ganglion cell, less action potential frequency, lower action potential frequency. So this slide, if you walk through it, explains how we can get an on-center and an off-center ganglion cell.
So sorry, why do you have the more of the first synapse one glutamate there on the left side?
We have the same amount of glutamate. We have an inhibitory receptor or an excitatory receptor. This negative sign is reflecting that this bipolar cell is expressing an inhibitory receptor. And this bipolar cell is expressing an excitatory receptor. The amount of neurotransmitter is constant. That is to say, in the dark it's a lot, and in the light it's less.
Like what defines off-center in terms of actual physical space?
An off-center is a cell whose action potential frequency goes down when light hits the center of its receptive field.
What these are just doing out.
They're arranged vertically. Let's go back to the cartoon of the retina. Here are rods. This is the center of these rods' visual field. Here is the surround of these rods' visual field. So center, surround. These rods are connected to these two ganglion cells via, it's not shown here, but via these two, let's just say these two bipolar cells. So here is the vertical, the linear organization. This is the center of this ganglion cell's, let's just say this ganglion cell's visual field. And if this is an on-center cell, then this photoreceptor was coupled to a bipolar cell that has an inhibitory glutamate receptor here. If this is an off-center ganglion cell, then this bipolar cell is expressing an excitatory glutamate receptor here. so now I'm gonna go screaming through all this, but my recommendation is don't do what I'm doing now. Walk through it and tell yourself the story with each click, what's, you know, maybe even predict, okay, what's the next click gonna look like? And see what, you know, see if you can do it. If you can walk through the story the way I just told it, you've got it. Brittany?
Are we responsible for the names of the receptors, or should we just?
No. Excitatory and inhibitory. You don't need to know that these are ionotropic glutamate receptors and this is a metabotropic glutamate receptor. Don't worry about it. Excitatory, inhibitory. Yes?
Can you just go through the excitatory
Go through what? The center?
The excitatory side.
Okay, go through the on-center
Yeah,
story?
yeah.
Okay. Let's go through the on-center story. So we're going to tell the story of this pathway.
No, do the other.
Is that the, I guess I'm getting confused. You're right, this one.
Yeah.
Okay. Light hits the photoreceptor cell, hyperpolarizes it. Less glutamate release. Hyperpolarization causes less glutamate release. Less glutamate, no, it's this one. Less glutamate activating an inhibitory receptor. Less inhibition is depolarization. Less inhibition is depolarization. So let's ignore the other one for a moment. Less inhibition is depolarization. More depolarization is more action potentials. In hyperpolarization, less glutamate release, less activation of inhibition. Less inhibition is depolarization. Depolarization, more glutamate release, more depolarization, more action potentials. Let's take a break. I'll leave this slide up. And then let's take a break and we'll get to what happens next. Let's take a short break. Okay. Alice made me remember that there's an important point that I meant to say, and that is that the photoreceptors are short receptors. The first cell in this pathway that fires an action potential is not the photoreceptor, it's the ganglion cell to which this photoreceptor is attached by a specific bipolar cell. So these are short receptor cells, and these are the cells that fire action potentials that go to the brain. The receptor cell does not fire an action potential. It has a graded hyperpolarization in response to light. Okay, so now we have converted light into action potential frequency in the optic nerve. There's the electrical signal going to our brain. Action potential propagation that you know a lot about. Optic nerve going to the brain, going to what part of the brain? Going to the lateral geniculate nucleus. So lateral geniculate nucleus is our next destination. Let's get oriented here. This is a superior view of the brain and the eyeball. So this is the left eye and this is the right eye because we're looking at it from the top. This is the fovea is right here on both eyes, and the blind spot is where these axons are going out of the eyeball. That's the blind spot. Each eye has a left and a right visual field. So you've got a left and a right eye, and each eye has a left and a right visual field. So I'm putting my fovea on Jacob, and I've got a left visual field, and I've got a right visual field. Okay, but my fovea is, I've got my fovea focused right there. Left visual field of both eyes, because I've got both eyes open, and the right visual field of both eyes. Okay, so that's what's illustrated here. Here's the left visual field of the left eye and the left visual field of the right eye, The right visual field of the left eye and the right visual field of the right eye. Right visual field, so I'm seeing my finger in, and it's hard to see because it's pretty far out in my right visual field of both eyes. Now it is in the right visual field of my right eye only. Left visual field of both eyes, left visual field, and I gotta bring it over here to see it. Left visual field, I'm gonna keep my eye focused on Jacob. Now I can see my finger moving out there in the left visual field of my right eye. It's out there, even if I'm not looking directly at it. You can play this game at home. You may, I don't want to say the wrong thing, so I'm going to say only the right thing, because what happens when teachers teach the wrong thing, that often is what students remember. I'm not going to say the wrong thing. I'm going to only say the right thing. And then maybe I'll come back and tell you the wrong thing later. But I'm going to only tell you the right thing. The axons from the left visual field of both eyes go to the right lateral geniculate. And the axons from the right visual field of both eyes go to the left lateral geniculate. It's not the eye, it's the visual field that matters in terms of left and rightness. So if the right visual field of the right eye is going to the left lateral geniculate, it has to cross over, and it crosses over in the optic chiasm. If the right visual field of the right eye ends up in the, if the right visual field of the left eye ends up in the left lateral geniculate, it doesn't cross over. The right visual field axons don't cross. So some axons cross. So we're looking at the right visual field. Some axons cross, and some axons don't in the optic chiasm. That's key. They don't all cross. Some cross and some don't. In order for the right visual field information to end up in the left lateral geniculate, and the left visual field information to end up in the right lateral geniculate. Now, to do this, you can see that the right visual field is focused on the lateral retina of the left eye, and the right visual field is focused on the medial retina of the right eye. So the lateral retina axons don't cross, and the medial ganglion cell axons do. And that's true on the other side too. The lateral ganglion cell axons on the right side that are receiving the left visual field, the lateral axons don't cross, and the medial axons of the left eye do. Okay? So you can remember it any way you want. But you don't want to remember what I'm not going to tell you. And you can probably predict what I'm not telling you, right? But I'm not going to say it because you'll tend to remember it. Remember that the left visual field ends up in the right, the information from the left visual field ends up in the right lateral geniculate nucleus, and information from the right visual field ends up in the left lateral geniculate nucleus. Or you can remember that lateral axons, that is to say, axons from ganglion cells in the lateral part of the retina, lateral to the fovea, end up not crossing, and axons from medial parts of the retina, medial to the fovea, do cross. It's the same information. So now, here's the same image from the father of neuroscience, Santiago Ramon y Cajal, who figured this out by just sort of thinking about, not just thinking, but mapping the trajectory of axons from medial and lateral ganglion cells to the lateral geniculate nucleus. So it was a brilliant inference from rather sparse data about how the retina is connected to the lateral geniculate nucleus. The lateral geniculate nucleus is organized into layers. I don't need you to memorize the organization of the lateral geniculate nucleus. So now I'm going to tell you stuff that you don't have to know. The lateral geniculate nucleus is organized into 6 layers, and information. Now remember, let me go back to the previous slide. There's information in the right lateral geniculate nucleus from the ipsilateral eye and from the contralateral eye, because both eyes have a left visual field. So information in the right lateral geniculate receives information from the ipsilateral eye and from the contralateral eye, and likewise on the left lateral geniculate. So there are layers, and the different layers of the lateral geniculate nucleus receive information from the different eyes. So just as a point of it, it's sort of interesting. Information from the ipsilateral eye, so if this is the right lateral geniculate, we're talking about the right eye, it ends up in layers 2, 3, and 5. And if we're talking about the right lateral geniculate, information from the left eye in the right lateral geniculate is processed in layers 1, 4, and 6. So there's just this interesting segregation of input in the lateral geniculate nucleus. Processing. There are different types of cells in the lateral geniculate nucleus. And now this is interesting because you know that you've got, your eyes do different things depending on where in the visual field the information is from. In my fovea, I am most interested in acuity. I want to be able to tell, when I put my fovea on Jacob, I want to be able to tell, that's Jacob, that's not Acacia. I can tell the difference between that input. When I look way in the distance, and I can see, that twig on that tree is not the same as that twig on that other tree, I'm doing high acuity vision. And high acuity vision is processed by a set of cells called the parvocellular cells in the lateral geniculate. But now let's think about the cells out in the periphery of our visual field. If I put my eye on Adam, I know that Mallory's face is over there somewhere, just from my memory. But I can't tell that Mallory's face is over there. I can see a face, sort of, not even. I can see a shape over there. But I can see Adam. I know that that's Adam. And I know there are people over there, but I can't really see them, and I certainly can't tell them apart. High acuity, low acuity. But if I move my finger out here, if I put my finger like right here, I can barely see it. But if I move my finger around out here, there's something there, I can see it. So the cells out here in my peripheral vision are good at motion detection. Our peripheral vision is much better at low light and motion sensitive processing. And this has an important evolutionary thing, right? If there's something coming at us in our peripheral vision, we want to know what it is. We don't even care what it is. We want to know that it is. There's something coming at us in our peripheral vision. Let me get out of the way, or let me divert my attention to figure out, is that a friend coming to hug me, or is that a bear coming to eat me? Okay, so we've taken information from the eyeball. Some of it is crossing over, some of that information is crossing over in the optic chiasm. Here's the lateral geniculate nucleus. Now we have optic radiations to the visual cortex. The right lateral geniculate nucleus and the left lateral geniculate nucleus. vision is going to be processed in the primary visual cortex next. But we do some other things with that information. So for example, some of that information goes to the superior colliculus, where our brain is working to keep our two foveas focused on the same spot. We want both of our foveas focused on the same spot. We don't want to be having one fovea going out here and the other, at least some lizards do that. They have, their eyes do that weird thing with one eye going over here like this and the other eye going over here like this. We don't do that. We keep both foveas focused on both spots, and our superior colliculus does that. Some of that information is going to the pretectum, which says, okay, is it a lot of light or is it low light? If it's a lot of light, we're going to send information out the oculomotor nerve to constrict the iris and limit the light. So the things that are not vision, orienting the eyes, controlling the pupillary diameter, other things like knowing how much light is coming in, is it day or is it night, controlling our circadian rhythm. These are all done by the retina, but it's not part of what we call vision. Vision is done in the visual cortex, and other things are done in other parts of the brain. But all the information, almost all the information, gets to these other parts of the brain from the retinal ganglion cells. Visual cortex, occipital lobe, calcarine sulcus. These are all names of the anatomical names associated with the primary visual cortex. Occipital pole, very posterior part of the occipital lobe, and along the calcarine sulcus. Calcarine fissure. Our visual field is organized in a map along the visual cortex. So here's a cartoon of the left visual field, right? I put my fovea on Adam, and I know that John is out here somewhere. So this is my left visual field, and this is my right visual field. So let's focus on the left visual field, which is going to be processed in which hemisphere of the brain? This left visual field is being processed in the right side. It went to the right thalamus, lateral geniculate nucleus, and it went to the right hemisphere, occipital lobe. So this is the right visual cortex. Occipital lobe, calcarine fissure. The ganglion cells that are closest to my fovea, so again, I'm staring at Adam, staring at Adam, left visual field, the axons, well, the left visual field is mostly processed in the right cortex, right hemisphere, like I said. The fovea is processed most posterior and more peripheral, so fovea is 1 and peripheral is 3 and 4. Fovea is 0, fovea, and most peripheral is 3 and 4. The upper left is processed in the gyrus below the fovea, below the calcarine sulcus. Upper left, right hemisphere, anterior, below. Lower left, right hemisphere, above the calcarine sulcus. So here is the map of the primary visual cortex of that visual field. Yes?
And so you see something that corresponded in the left primary visual cortex that would correspond to the right.
The right visual field, that would be stuff going on over here.
And so you have the stuff in the upper right quadrant and the lower part of
Right.
the left.
So the points to make, the things to learn here, are the fovea is most posterior, and the more peripheral is less posterior. Fovea is most posterior in the map. peripheral is less posterior. Lower visual field above the sulcus, upper visual field below the sulcus.
Why is everything reversed like that? How left and right go from?
You know, I don't know. Not enough of an evolutionary biologist to be able to explain how we got from like barnacles that have photosensitive, or like clams that have photosensitive scallops that have those eyes sort of along the edge of the clam, along the edge of the shell. So they have photoreceptors. We evolved. We have a common ancestor with the scallop that I don't know how it evolved that way. The information is processed in the occipital pole, right? More occipital, more fovea, less occipital, or less posterior, more peripheral. And then we divide this, now we're talking about real vision. We have a stream of information that goes from the occipital lobe towards the parietal lobe, and that's asking, where is this thing? And we have a stream of information that is going from the occipital lobe into the temporal lobe, and that's asking, what is this thing? So in my peripheral vision, I can't tell whether that's someone coming to hug me or a bear coming to eat me. But if I put my fovea on it, now if there's something coming at me, I'm using my dorsal stream to determine where is it, but I don't know what it is. And then I put my fovea on it, and I say, oh, that's a bear. It's coming to, I gotta do something. I'm not gonna wait for its hug. I'm using my ventral stream to determine that that's a bear, and that's dangerous, and I better respond, and not just by opening my arms and accepting the bear hug. Okay, so dorsal stream, where it is, ventral stream, what it is. So let's end this with some, so there's some links that you can use online, but let me just do a few optical illusions. These are sort of fun. So you're now using the information that, so I don't know if you can see, but this is the grid illusion. If you put your fovea on the white spot in the center, at least what I see is a bunch of those other spots look dark. They look black. But if I put my fovea on, so if I look at this one, my peripheral vision says this one here is black. I'm putting my fovea here, but I'm using my peripheral vision to see the spot over here, and it looks black. But then when I put my fovea here, it's white. So is it white or is it black? Well, it's white because your fovea is giving you the best acuity, but your peripheral vision is giving you some misinformation. And especially if you move things around, the white dots turn black, and right? Are you all seeing it? Yeah, this is a classic visual illusion. We see edges. That's what we see. We see contrasts. We don't see absolutes. So it will surprise you to know that this color gray and this color gray are exactly the same color. What? This color gray and this color gray are exactly the same color. Let me prove it to you. I'm going to put up, all I'm doing is adding a blocker. So I have not changed the image. I have promised you, I have not changed the image. I have simply put a mask over it. And so now you're saying, okay, now I believe this gray is closer to this, but it still doesn't look exactly right because your brain is knowing, oh, this is up, there's the sky, and this is down, this is the ground. All I'm going to do is flip this upside down. Now you believe me that this gray is exactly the same as this gray. What our brains do is our brains figure out, oh, this tile is white, so this gray must be lighter than this gray. That's our brain interpreting, not misinterpreting, that's our brain interpreting the image. But the grays are exactly the same. And here's another example of this. You can go to these websites if you're interested. This tile and this tile are exactly the same color. The gray of the A tile and the gray of the B tile are exactly the same intensity of gray. Your brain, go to the websites and it'll prove it to you. Your brain is interpreting, oh, this one's in shadow, so this must be lighter than this. That's your brain interpreting the information to your eyes. Your brain is creating the image out of edges and contrasts. And here's another one. So this is the last one. Vision is a mental construct. You see, in some cases, you see what you expect to see, or you see what you've been trained to see, or you see what you're used to seeing. So I'm going to remove the mask in just a moment. But children under the age of about 8, in the image that you're going to see dolphins. Adults, the rest of us in the room, see something else, and you'll see what I mean in 1 second. Children see dolphins. Adults see an image of 2 lovers. Can you see the dolphins? I mean, there's a dolphin, and there's a dolphin. Children only see the dolphins. They don't see the naked humans. So our vision, what we see, is adjusted by what we know. So vision is a mental construct as much as it is we're not cameras. Okay? And so now I'm not going to have time to start the auditory system, so I'll start the auditory system on Tuesday.